Myself, I'm Dr. Lee, and with me is my colleague, Dr. Timothy Tan. We're happy to share with you about hands-on and minds-on learning of science using a microbial fuel cell. Timothy was deeply involved in this project, which we applied for some years ago. So maybe I asked Tim to share with us, and he will walk us through some of the findings and the details of the project. And after that, please feel free to ask questions. We're happy to share what we know about that. Thank you.
Right, thanks, Eugene, and welcome everyone to this session. Okay, let me just swap over. Now, it may be helpful, especially if you're in gallery view right now, to switch to speaker view. So because I'm going to have the slides behind me in my own window, I'm not actually sharing in the traditional way. So it's just going to be behind me. So if you switch to speaker view, it should be fine. Unfortunately, in a breakout room, we cannot spotlight the video and sort of force this for you. So, okay. So let me just start by saying that this project was done several years ago, and it's sort of an ongoing, on-running thing that we've been working on. What I'm going to be sharing with you is some of the findings from the study that was actually done a few years ago. I'll just move along very quickly to say that when we are talking around this idea of inquiry learning using a microbial fuel cell, or I'll just refer to it as the MFC, right? The microbial fuel cell. It started out with from a centered around interest around how to make inquiry learning minds on. Because this comes from the common observation that a lot of science practical activities, and even those that are intended to be inquiry driven or inquiry learning, tend to be very focused on the hands-on and not always enough attention paid to the minds-on aspect of it. In other words, one student may very well be very engaged in practical work, but may not actually be learning anything, right? So very often you see findings such as, great, you did a water rocket or you did a egg drop experiment, but when you ask the student after that, what did you learn from it, or how does this link to something that you could have learned? They just shrug their shoulders and say, don't know. Did you have fun? Yeah, it was fun. Great. You know, we had a good time. It was so engaging, but what do you learn? Don't know. Right. And even if it's not about metacognition, testing the students and all that, they don't always, there's no clear link, right, for them very often to what they're supposed to be trying to learn or they're supposed to be learning. So I think we started out with this from this standpoint, and we say, how can we make learning much more hands-on? So insofar as inquiry learning approaches go, I think project-based learning, problem-based learning, and design-based learning are very often cited as different approaches to this, and quite often confused, right, or conflated. So just want to very quickly say that the area that we're working in is this thing called design-based learning. And design-based learning is quite often described as being part of problem-based learning, and problem-based learning is part of project-based learning, and project-based learning is in this bigger area of inquiry-based learning. And we'll sort of use this idea that it is all of these things. So design-based learning is a very specific subset of all of these areas, including this big umbrella of inquiry-based learning. Okay, so what is design-based learning? Basically comes down to what we refer to as design-based inquiry, to foreground the word inquiry here. So DBI is basically design and make. So it's relatively familiar in a lot of examples, such as I've already mentioned, egg drop, water rocket. Designing a paper aeroplane that will, or making a paper aeroplane that can fly the furthest. One thing that is common with most of these design-based inquiry activities is that they tend to be very focused around, or very centered around, physical science type learning activities, right? So engineering and physics being the foreground topics to be covered, subject areas to be covered. So it's logical, because when you're talking about making things, you tend to be thinking of making physical objects, and physical objects, usually the easiest thing to test is their physical properties. So design and make activities, DBI, this is what we are essentially talking about. one of the problems is that, as I mentioned, many of these things tend to be quite engaging, interesting, get a lot of student excitement and interest going. But, why did your paper airplane fly at the furthest? Or they will give a lot of explanations that may not always necessarily tie into the physics that they're supposed to be learning. For example, the physical sciences they're supposed to be learning. Why did your egg drop survive the thing? Nothing about force, acceleration, momentum, Young's modulus of elasticity. None of these things ever come out. They always come out about, I don't know, I just packed it full. So this is where the minds on issue comes about. And so to try to force that, we needed to find something that would make students think about the science and where possible, unless you think about the science, you can't get very far with the design and build, right? Or design and make, or to be very successful. Because if you randomly try stuff, then that's one way of doing things. But in the interest, in the service of science education, we want students to be thinking about the science of it, right? So, and by extension, actually, I will broadly be speaking about how this fits into STEM education, because there is the math in some of these things. There is the engineering and there is the technological learning in a lot of these kinds of things. But largely, I will focus and use the word science as the general topic, because that's the main thing that we could be learning from this. So what could we do? So one of the things that I quite early on was very interested in, I've been interested in for 15, 16, maybe or more years now. It's been this thing called the microbial fuel cell, which I will just refer to as the MFC. So the MFC is something that is very interesting. You put in a microbe, obviously, bacteria, it could be yeast, it could be algae, something that is in that sort of unicellular form, and you put that into the cell, and basically you get electricity out of it. And this sounds like a science fiction concept, right? So if you remember the Matrix series of movies, that idea of a living thing as a battery, as a power source, is not actually very far-fetched. The similar principles, not exactly similar principles have been applied to snails and plants, and you can get electricity because at the end of the day, electricity is one form of energy. And as living things, we consume food in order to transform that food into energy. And from a biological perspective, and from a science perspective, that energy largely comes in the form of electrons. So you don't think of it that way, but the energy in respiration, oxidative phosphorylation, and so on in cellular respiration, the energy processes there basically generate electrons, and electrons are the currency, the ultimate energy currency. We talk about ATP, but ATP generates its energy currency or exchanges energy ultimately in the form of energetic electrons that are being exchanged from molecule to molecule. A broad class of chemical reactions that we refer to as redox reactions, reduction oxidation reactions. So a series of reduction oxidation reactions are what powers the cell, right? Any living cell. But redox reactions are predicated on the fact that they are passing along, at the end of the day, electrons. So if that sounds like the concept of electricity, well, it's similar, but through a process of biology to chemistry, so biochemistry, redox chemistry, and then electrochemistry to physics, the passing electrons to the external circuit. I won't go into the mechanics or the details of this, but electrons from the living cell in its breakdown of food through respiration gets transferred to external circuit through biochemical and electrochemical means. Okay, so it's a very sounds like a convoluted concept, but basically you add yeast, you add some chemicals, you get electricity coming out of it, and of course you feed the yeast with sugar. Okay, so that's the essential things you need to know. Now, why is this cool? Is because it's biology, chemistry, and physics all rolled into one topical area, one set of practical activities that require understanding across these three sciences. primarily chemistry, the heaviest aspect is chemistry, but you should understand something about biology, understand some basics in physics in terms of electricity, circuits, and so on. That's what makes it great. You can integrate across these three sciences. And so that's what was the primary interest from years ago when I first got involved in this. And of course, in time, the idea here is that energy sources, alternative energy sources, and the sense of purpose that you heard earlier, right? That Mark and Dennis were, I think Mark especially was talking about, the sense of purpose that you give students, because they're very interested in environmental issues. They're very interested in alternative energy and clean energy and green energy. So the MFC provides a context in which students are very engaged in, very interested in. And that's why it's particularly useful. So for all of these reasons, the microbial fuel cell is a particularly useful inquiry object, center for inquiry activities. Okay, so that's it. Okay, there's something wrong with the slides here. Right, so the design features of MFC very quickly were then that we give them authentic scientific experimentation. So it's not something that's textbook. They have to actually engage with it to do it. Integrative science, as I've mentioned, there's STEM integration because there's an engineering design challenge built into this, and a lot of applied science. And ultimately, we hope, when we did this project, to achieve science education in what is described as science education in three-part harmony. So I'm going to get to that later, talking about what this three-part harmony is. But I want to highlight also that because of the nature of the scientific, the way the micro fuel cell works, there's a lot of scope for authentic scientific experimentation. You can substitute parts and change things and change reagents and change concentrations, change time over duration. We don't often think about examining outcomes over time. So we tend to do science experiments as endpoint experiments, right? What is the endpoint? What's the change in pH at the very end? What's the temperature at the very end? And things like that. The temperature changes. So we don't think about what's the voltage change over 24 hours, over 1 week, things like that. So these are some of the things that we can possibly do with it. So the microbial fuel cell challenge was therefore conceived as something where you design and make your own microbial fuel cell. Once you understand a little bit about it, you learned a little bit about it, gives you collaborative small group work. There's a design-based challenge, that's DBI, there for you, as well as a little bit of friendly competition. So this idea of inquiry, competition, challenge, and collaboration, I think brings about a very strong motivation to learn. And that's what we're looking for. So very quickly, I just want to outline what the program was. The program that we did was with secondary 2 express students. Typically, we were working with the high progress science learners in the schools we worked with. And typically, it's about a 10-week program. 10 weeks doesn't have to be continuous. It could be broken up across 2 terms and things like that. Holidays and everything taken into account, and so this tended to be spread out. So sometimes the content upgrading was done at one point, and then the main bulk in the middle was done somewhere, and then the tail end was done a few weeks after that because of whatever reasons. So the thing with the nature of this is, of course, we did require multiple teachers to be involved. Nominally, we need a biology teacher, a chemistry teacher, and a physics teacher. And of course, it'd be great if one teacher was straddled more than one subject, but it's ultimately something that needed to be team taught. And we got the teachers themselves to enact this program. So content upgrading was just bringing Sec 2 students up to speed. There are certain things that are not covered in Sec 2 syllabus, ions, stuff like that, that would be helpful to understand or not yet studied. So these were teachers brought the students up to speed over the first couple of weeks. Then we got them through guided inquiry. And this is probably the most familiar aspect, science practicals, and yet still very different, because what we actually required them to do was to take a data logger, voltage sensors, and then do experimentation using control conditions, right? So control conditions using kit microbial fuel cells. So these are purchased microbial fuel cells that can be assembled in a very fixed, reproducible way. And you are detecting the voltage output, changes in the voltage output when you change a condition. So if I use a different sugar, or if I used more yeast, or if I change the chemical compositions within the different chambers of the fuel cell, would I get a different result? And you'd be able to trace different voltages there. And we did this iteratively. So the first week was very, very guided. We assigned what different groups were supposed to do, such that they then collectively contributed results back to a common pool. Students would then examine the data from that pool and then come to a decision as to what they wanted to investigate the following week. And so we progressively withdrew the scaffolds. The second week was semi-guided. And by the third week, students had to come up and design their own experiments. They had to decide what they wanted to do, put in a requisition for the chemicals that they wanted from a menu that we gave them, right, of what they wanted. And so they progressively got to design their own experiments. The third part of this was where the actual DBI came in, the design-based inquiry. And the design-based inquiry came in with this idea of this iterative design of prototype. So there's an engineering approach to this, there's engineering design required. But the key thing that drives the design-based inquiry is this idea of iteration. You make and you fail. So mathematicians, when you're familiar with the idea of productive failure, and you will have to, in a sense, fail to learn from it, right? Experience is the name we give our mistakes. And so students start out by drawing their designs. We sort of vet and check and give advice, but we want them to build. So they start with the everyday materials. We tell them, bring stuff from home, make stuff. So again, the upcycling mindset here comes into play. Students very much are very interested in this idea where they're taking, making use of their things, but they build stuff that typically first try doesn't work very well, right? It leaks, it doesn't hold, it doesn't seem to produce a very good voltage at all. And they're making stuff that falls apart, but they learn from it very quickly. And they eventually realize, oh, we should use plastic, or we should use super glue, or we use the epoxy glue that's provided to us instead of just tape and cardboard, right? So things get better, and you can therefore trace that they're learning things, right? They're learning things through doing. And it's not a random thing. We're kind of forcing them into this cycle of planning, making, evaluating, and then going back, right? Okay, with what we've learned, can we apply to this? And over time, ultimately, we get to the point where we have a set point that a certain week, a certain time, we're going to have a challenge. And that challenge is going to be, you design the most powerful microbial fuel cell you can. Powerful in inverted commas, powerful in terms of voltage, in terms of possibly current, in terms of number of LEDs or light up, in terms of longevity of the cell. And what happens is that when we give them these different design challenges, different groups of students may have differing goals. They may say, we're going to try and produce the highest voltage. Some may say, wait, if the highest voltage, and then, by that point in time, from the theory that they've learned, from the hands-on experience that they've got, they realize, they slowly begin to realize that sometimes different design goals have differing needs, right? So if you want to have high current, it's a different design from having a high voltage. Having longevity requires a different set of again, parameters. So to give you a brief idea, high voltage may mean using certain chemical setups, waiting a certain period because the voltage climbs over a certain time. So you need to set up your cell 2 hours in advance to reach the peak voltage. But you want a small compact cell relatively because you want a minimum kind of resistance kind of thing. Now, students don't always understand all of these things, but they know that they want large surface area of electrodes and so on. Whereas longevity doesn't require that. Longevity may actually require large volumes of chemicals, or a larger volume, more yeast cells or more sugar or something. So these competing things give students room to argue for their design. So there's a lot of built-in argumentation, using of evidence from the prior experimentation, from prior week's builds. They learn, okay, we need to improve our design, but do we go for larger surface area or larger volume? And these are competing design criteria that students then argue out, right? And then they try to find evidence to gather support on their side. So in teams of 4 students, they will vote on what they want to do, right? They usually, and they kind of naturally fall into this. They end up using the science to argue their case, which was very gratifying to see. So where does this fit into this whole idea of learning would be the fact that science education could be described by a paper by Professor Dushel, is this idea of scientific literacy as being composed of conceptual, epistemic, and social learning goals. Now, science education, especially in Singapore, has always tended to favor conceptual learning. The most you learn in epistemic is this idea of procedural skills, right? Procedural, or what we often refer to as process skills. But epistemic learning goals go beyond that. Epistemic learning is knowing why we know what we know, right? So that's the nature of science, the nature of science as being evidence-based, as you know, and so on. And social learning goals is this idea of being enculturated in the processes of science, that science is a collaborative effort. It's a collective endeavor, that you need to prove your case, and there will always be questions about from peer critique, right? Peer review that you're getting. And those are some of the elements of social learning. And we don't do enough of this. But the nice thing about the microbial fuel cell is that actually the conceptual learning goals could be almost said to be the minor aspect. The learning in science is relatively minor. It's a bit scattered. It's the problem with the microbial fuel cell is admittedly that and it's hard to convince schools to take it up because the LOs that map to the syllabus, right, the learning outcomes, tend to be scattered across from lower secondary to Sec 3, Sec 4 to even JC level science. So bringing them all together in one level is kind of difficult to convince teachers to do. But I will point out that we got secondary 2 students to very successfully learn a lot of these things. But I think the greater learning was in the epistemic and social learning aspects, as I will bring out very quickly. So I should have switched to this slide to point out what these aspects of conceptual, epistemic, and social learning goals I was referring to, what I mean by these things, right? So a lot of this thing, application of knowledge to decision making, was something that we found was a very critical thing that needed to be done. Because it was iterative, because of iteration, students saw for themselves, hands-on, this is the results we got from last week, or this is what others, teams, other groups in the class got from the previous weeks. And how does this inform our decisions, our design decisions, our experimental decisions for this week? What are we going to do this week? How are we going to do it? Well, last week they found that this sugar is better. Another group found that this other sugar was maybe better. So which do we do? You know, sucrose or glucose? And they will kind of have to make decisions based on this. This is so different from current science practical practice, where this week's science practical has nothing to do with next week's science practical, right? Science practicals tend to be very standalone activities. And you therefore don't give students the opportunity to engage in this process of looking at evidence and making decisions, what we call evidence-based decision making, or even evidence-based explanations that they have to come up with to support their decision making. So all of these things that the MFC program enabled students to do. Scientific literacy, I think in the interest of time, I'm not going to talk too much about this, but we did think about not just in learning terms, in learning goal terms, as I've just said, but in terms of the competencies. And these are all in the literature anyway, that we wanted students to be able to develop these competencies. And we did see evidence of students being able to do all of these things. Okay. So where does the MFC come in is that in terms of developing scientific literacy as an educational goal, the focus of STEM integration to provide a context, a real world, minds-on kind of context for students to engage in. And this idea of design-based inquiry as the pedagogical approach that forces students to really think about all of these things. That's where the MFC sits, right? At the confluence of these 3 major areas. And the really exciting thing to see basically is that students were able to do it. Secondary 2 students were able to do this. They could build progressively better models. There's leakage. You can see actually in all these photographs that there's a lot of leakage of the chemicals, right? They learn from it. And it's actually a really hard thing, right? You can get undergraduates to try to design something and they don't necessarily do any better. In fact, in many ways, in a few instances, we saw better learning in Sec 2 students than we saw in undergraduates. And I mean, we didn't delve very deeply into it, but just off the, one possible way of interpreting it is that maybe, the higher you go, the more narrow focused you are in your mindset. And this idea of integrating across disciplines, looking across disciplines was very difficult. Mind you, actually, the students really had the easiest time. Secondary school students are still very malleable. They're still very open to things. The hardest group, honestly, was actually convincing teachers. And we often saw this. We saw teachers who said, I'm a biology teacher, I don't know anything about electric circuits, right? And then you have physics teachers saying, I'm a physics teacher, I know nothing about respiration. When all of these things are covered at primary or lower secondary level before. So as a science teacher, you did science in school, you would have covered these things. You cannot say you don't know what a series circuit is or parallel circuit is if you didn't do physics. You should know what it is. So it's actually hard because this sense of teacher identity and your disciplinary identity tended to kind of override and cover this up. But we did see that students were capable of learning. So it's entirely possible. Not only that, some of them produce some really, really powerful cells. Now, this one here in the upper right-hand corner, I think ended up being one of the top ones, something like 0.8 on 0.9 volts. Now, 0.8 or 0.9 volts sounds terrible, doesn't sound like much. But given that the standard cell produces only about 0.5, that means out of the teaching kit, if you buy the kit ready-made and everything, you'd only start with about 0.5 volts. So student experimentation leading to coming up with stuff that leads up to about 0.9 volts is a fantastic outcome. They're really producing. We're producing increments. So if the students go from 0.5 volts to 0.55 volts to 0.6 volts, that's wonderful. They go to 0.75 volts. Wow, that's a really excellent outcome. But we actually saw some, really fantastic efforts. I mean, obviously this is a gigantic MFC. It's huge. It's like 1 liter in each chamber. That's, crazy, but they managed to get it to work the way it did. So where did student learning come from across these 3 domains? In terms of skills, we can put it this way. These are the blue boxes. And in terms of the conceptual learning, sorry, not conceptual learning, but the epistemic and 21st century competencies that you can refer to, these are areas where we got students to basically enculturate as student scientists. They tended to then talk as if they were, putting forward things, and then they were arguing with each other about the evidence, and they were trying to think of themselves actually as doing actual work. In fact, we so convinced them of this, they were excitedly asking us about whether their designs can feed forward and whether they'll solve the energy crisis kind of thing, just because they've found an MFC that really was more powerful than the standard one. So I mean, that was really interesting. I mean, a little bit naive for the students because they really thought that they were, but that gave them that sense of purpose that they felt really was very, very powerful. And of course, we didn't really disavow them of that idea, like, yeah, definitely sure that this is really a breakthrough finding. And the funny thing is that a lot of these things are not actually published in literature. So in many ways, a lot of the experimentation they did and the particular combinations of chemicals, the concentrations that they found that were needed, were actually, in that sense, scientific breakthroughs, because they're not actually known published results, right? They're not fantastically, groundbreaking in a bigger scheme of things, but insofar as what they were doing, they were not going through textbook knowledge. They were not, just working on something that was already there. So they enjoyed the lessons and they did really well. They really came out with some really fantastic things. So in the last just couple of minutes that we have, I just want to bring up this idea that students have learned a lot of things in competencies. Particularly, we're very, very encouraged by the fact that they were able to use evidence to reason with and make decisions. They could plan their own experiments and deal with the very messy data. Data wasn't clear or clean. There wasn't linear relationships between concentration changes. There were very, very peculiar curves, right? Including very counterintuitive results. But they were backed up by the fact that 8 different groups got the same result. So it's not a anomaly. It's the way it is. And how do you explain these results? What's an inverted bell curve mean to you? Inverted bell curve to a scientist is kind of weird, right? you'd normally expect a bell curve to look like this. And here you're seeing a U-shaped curve when you vary concentration of a particular reagent. Voltage output is a minimum at a middle value. It's normally optimum, right? It's the Goldilocks idea of there's a nice medium middle ground, but this wasn't the case. So these are some of the great things. In terms of social domain, as I've already mentioned, very self-directed learning, very motivated learning. Students actually felt a lot of agency because we gave them the agency, the authority to do their own things. So they were really excited about that. In many cases, students began to be, not all, but many of students began to be self-aware of their epistemic stances, that they realized they were coming from it from a very science-based perspective, or very sort of just get it done engineering perspective kind of thing, like going for optimum as opposed to trying to understand the big picture. They were also quite aware of the learning that they were, they wrote a lot about, wow, I learned about this, and it's so great to be able to apply the science that we learned. Now I see the big picture, and now I see why we learned this thing in science, So those were really, really encouraging reflections from the students. There were, of course, issues. Inquiry-based learning generates all kinds of issues. One of the things about inquiry-based learning is that success tended to require a diverse group. And actually, this is for other areas of research. We know that diverse team makeup is very important. If you have everybody thinking the same way, you don't have very high success. Or you have very extremist, bossy students that also they work very well. And of course, I think these are logical and come from elsewhere as well, understandings elsewhere as well. So very, very quickly, development of a science curriculum, we successfully implemented it. We figured out what works. It's a model for development. And let's see. Yes. So I think we developed, we were on track to produce authentic scientific literacy. Okay. One more thing is, yeah, students were very effectively engaged. They really, really were very turned on by this. So just to say that we acknowledge the grant funding from OER and a school from the East Zone, a government-aided school that really helped us in developing this whole program, because the teachers were very actively involved in co-developing this. I think we have 2 minutes or minute and a half for any questions. Anyone?
Okay, one question. How do you adapt this for primary science context?
It's entirely possible. We just take out a lot of the detail, right? You just suck it all out. We just give students with the experimentation of here's reagent A, reagent B. Limit the choices. Make it simple. If I try this and this, what's the voltage? If I try this and this, what's the voltage? And then get students to try to begin to understand it. We don't feed them too much of science, but it's entirely possible. I feel that the microbial fuel cell is something that, because it is something that you can almost do with kitchen science. There's a lot of chemicals can be substituted for kitchen science. You can actually just use cellophane paper, bag it up, put it in a container, put baker's yeast. In fact, many of the chemicals we're using are just basic stuff, sugars, baker's yeast, potassium permanganate, which you can get from a Guardian pharmacy if you wanted to, right? These kinds of things are all there. So yeah, okay. So the admin says we're going to close the rooms. By all means, if you have more questions beyond this, you can always email me. I'm just timothy.tan@nie.edu.sg. Let me just put that in the chat. So if you have any detailed questions or you want to find out more, feel free to email me. I'll be happy to try to answer your questions.
Yeah, in fact, okay. Just before lunch, Timothy and I were discussing about fruit battery scenario. So some schools are having some issues with fruit battery, and it's something like a design-based, and but it's actually quite interesting a scenario to work with. So MFC, electricity, fruit battery is also another side, very closely related kind of project that primary school kids can try.
9 seconds. So the room's about to close.
Sure.
Yeah, thank you everyone.
Thank you everyone. Yeah.
Thank
See you
you.
back.
Thank you.